Well, thank you all for being here today. So my goal today is to go over some new approaches to engineer conditional alleles in mice. And this is a technology that we have developed recently, well, over the past several years, actually, but we're publishing this paper should be accepted, should be published pretty soon on this. So the technology involves the use of short artificial introns to engineer conditional alleles. So, like Jill just mentioned, my name is Stéphane Pelletier. I'm the director of the Indiana University Genome Editing Center at the School of Medicine here in Indy. So before I get started, I just wanted to give people a short update on our facility. These numbers are since the inception of the core in July 2020, and we've completed the generation of 18 mouse models. And we're currently working on engineering 15 more models. So if you're interested in engineering mouse models, you can reach out to us and we'll be happy to help you out with this. We've also completed 38 cell line models, and we are currently working on 21 additional projects. On the transgenic side of the business, our facility stores more than 310 cryopreserved mouse models. We have performed more than we have performed 6 pronuclear injection services. We have cryopreserved 180 lines over the last 2.5 years. And we have performed 36 in vitro fertilization or rederivation of mouse models. The core employs 5 full-time employees, 1 part-time employee. We have 1 collaborator, which is Judy Hallis from the Purdue Transgenic Core. And we are currently recruiting for 2 other positions. One is a lab research assistant position, kind of entry-level position to come and help us engineering cell lines and stem cells. And the other person is a microinjectionist who should join us, but not a done deal, but he should join us in June next year. All right, so as you probably know, genetically modified animals are the cornerstone of research programs across many fields of study, including anywhere from immunology to cancer biology. Our understanding of the genetic mechanisms of human diseases has been largely expanded by the loss of function studies using engineered mouse models. There are 2 types of gene knockout mouse models that are commonly used. The first one is what we call a global gene knockout. And the second one is the conditional gene knockout or conditional alleles. So the global gene knockout refers to the ubiquitous deletion of a gene from all tissues of an animal. Global knockout models can mimic the genetic condition of human diseases, therefore allowing a quick and thorough evaluation of gene function in vivo. The design of these models is relatively easy and can be done by introducing frameshift mutations or via the targeted removal of 1 or more exons of a gene. However, the genetic studies using this type of model may have some limitations, I would say, or inherent limitations. For a gene that is widely expressed and pleiotropic effects from its deletion in all tissues can obscure the cell type specific gene functions. And the second limitation is that an early onset lethality or gross abnormality of a mutant will prevent, of course, its application for studying gene function at an adult stage or during aging conditions. So to get around these 2 main limitations, people have developed new strategies. One of them is conditional gene knockout, and this refers to the selective inactivation of a gene in a spatiotemporal manner. The main advantages of this approach is that site-specific recombinase system can remove sequences flanked by 2 singles site-specific recognition sites through DNA recombination, thereby remove parts of a gene to inactivate it. Several systems exist. The most common one is the Cre-LoxP system. There's also the flip Fritz system, and there's a new system called Vica-Vox, Dre-Rox, Negri-Nox, and Pentopox. There are several SSR lines that have been engineered that allow tissue-specific expression of the SSRs, or for a Cre-recombinase, for example. And there are more sophisticated models also that exist, those that are hormone sensitive or can be induced by tetracycline treatment of the animals, if you will. So there are many models that exist, many systems that have been developed, and these models have afforded valuable opportunities to integrate the context-dependent gene function, thus providing clinically relevant information to treat genetic diseases. The major disadvantage of these models is that the generation of these alleles is very complex, it's time-consuming, it's tedious, and very, very, very expensive. There are two main strategies that are currently employed to engineer conditional alleles in mice. The first consists of flanking a gene or critical exons of a gene with site-specific recombinase sites like LOXP sites, as shown here on the left-hand side of the figure. And these LOXP sites are positioned such that upon Cre-mediated recombination between the two LOXP sites, a critical exon or an entire gene, for example, can be removed, resulting in the generation of a frame shift and generation of premature stop codons. The second strategy employs actually large artificial introns. There are two systems. One is called conditional by inversion and is shown here in the middle. And the second one is CRISPR-Flip. And the way these things work essentially is that you knock in a large artificial intron that contains sequences encoding either reported genes or anything. And these genes are running in the opposite direction of the gene, such that upon Cre-mediated recombination, these cassettes are flipped, exposing a splice acceptor site right here. And this splice acceptor site promotes the splicing of exon 1, if you will, part of the exon 1 into this splice acceptor, and allows expression of the reported gene, but prevents expression or knockout the expression of the endogenous gene. CRISPR-Flip works pretty much the same way. Oops, I'm sorry about this. Works pretty much the same way. The system is slightly different. These systems can be used to actually also express reporter cassettes or reported genes. So that's kind of an interesting feature. So both of these systems can be engineered using either CRISPR-Cas9 injected directly into zygotes, or facilitated, or they can be used to engineer mouse models by conventional ESL gene targeting. So while these methods actually have been used for more than 2 decades and have been used to engineer like thousands of animal models with conditional alleles, these methods are, the flanking of one or more critical exons with SSRS in zygotes, for example, it cannot be used to, there are some disadvantages associated with each of these technologies. And one of them is that flanking one or more critical exons with SSRSs or site-specific recombinase sequences in zygotes cannot be used to inactivate single exon genes. This technology does not allow for the expression of a reported gene upon Cre-mediated recombination of the allele like the other two systems. And the success rate of inserting 2 SSRSs on the same allele is actually very low. It's less than 10% of the mice that are produced will have the 2 LOXP sites, for example, inserted onto the same allele. So because of these limitations, it oftentimes the generation of these models require multiple rounds of injections, and this is costly, and it uses many animals. In the same way, flanking one or more critical exons with SSRSs using gene targeting in ES cells, it cannot be used to inactivate single exon genes. This method uses drug selection cassette that must be removed in a second step to ensure proper expression of the targeted conditional allele. This technology makes use of large and complicated targeting construct. Oftentimes, these constructs can be anywhere near 30 to 40 kilobases in size. So we have a special backbone plasmid actually to amplify those. The technology requires gene targeting in ESL and production of chimeric animals. So this adds up to the complexity of and the success rate of engineering conditional alleles using these technologies. And oftentimes, this technology requires multiple rounds of injection, obviously, to generate chimeric animals that are going to be capable of transmitting the mutation via the germ cells. So conditional by inversions. These, like I just mentioned, they use drug selection cassette that must be removed in the second step to ensure proper expression of the targeted conditional allele. So all of these are used usually to engineer mouse models using conventional gene targeting. So we have to have selection markers in there to facilitate the identification of clones that are properly targeted. This technology makes use of a large and complicated targeting construct, a little bit like the conventional gene targeting or fluxing exons with the conventional method. This method requires gene targeting in ESL and production of chimeric animals, like I just mentioned. Oftentimes, it requires multiple rounds of injection, and reporter alleles can affect expression of neighboring genes. So essentially, when you start introducing larger DNA construct with strong promoters to drive expression of a bunch of genes, these promoters can actually affect the expression of surrounding genes. And this has been reported in molecular cell biology not too long ago, a couple of years ago. So that was kind of an interesting observation. Finally, CRISPR-Flip is also typically used in the ES cells. It makes use of large and complicated targeting constructs. The technology requires gene targeting in ES cell, like I just mentioned. Sometimes it may require multiple rounds of injection, although the use of CRISPR actually facilitate or simplify the process because it is somewhat so efficient. Reporter alleles can also affect the expression of these neighboring genes. So I've been working on engineering conditional alleles for quite some time. So I've had tremendous success, but we also face some very poor rate of our rate of engineering those alleles actually was actually pretty poor. So to get around these limitations, if you will, and improve our ability to engineer conditional alleles in mice, we turned to a recently described system that is called a degradation based on Cre-regulated Artificial Intron. And in this system, a short 201 nucleotide long cassette containing intronic sequences is inserted within the exon of a gene, and that's the schematic representation of this here. And this on top here is the sequence of the short cassette. So the cassette essentially contains sequences encoding a splice donor, as shown here in green. This is followed by a LOXP site. Intronic sequences, including a polypyrimidine tract, as shown here in yellow, a second LOXP site, and then a splice acceptor site, as shown here in red. So this cassette is inserted within the exon of the gene. And so in the absence of the Cre recombinase, as shown here, the cassette is recognized as an intron by the splicing machinery of the cell and is removed. This allows the normal expression of the gene. So the entire cassette here is going to be spliced out under normal conditions. In the presence of the Cre, recombination between the two LOXP sites removes essential intronic sequences. So there will be recombination between the two LOXP sites here, removing these the branch point in the polypyrimidine tract. And what that does is essentially this small cassette now will no longer be recognized by the splicing machinery. And this will leave a string of premature stop codons within the exon of the gene. And this will inactivate the allele. And it will send, at least we thought, that it would send the mRNA transcript for degradation via the nonsense-mediated mRNA decay pathway. So we wanted to know whether we can use this this approach to engineer conditional alleles in mice, and we decided to insert this cassette within exon 3 of the SCYL1 gene, as shown here. This is the SCYL1 gene, and we wanted to insert this little cassette within exon 3 of that gene. And so, and we did this essentially by using CRISPR-Cas9 technology, and we chose exon 3 of that gene essentially for two main reasons. The first is that the presence of a premature stop codon within the 5' half of a gene and within the 5' half of an exon. So essentially, the positioning of that cassette it does matter in order to promote mRNA degradation via the nonsense-mediated mRNA decay pathway. So you cannot introduce a stop codon anywhere in a gene or anywhere within an exon. It has to be to promote nonsense-mediated mRNA decay. The premature stop codon must be placed early in the gene and also in the 5' half of an exon. So this way, you can increase your chances of inactivating the allele this way. And the second reason is that we actually surmise. So we have no proof for this, but we surmise that that inserting the artificial intron within exon 3 of a gene, which is the biggest or the largest exon of the gene, we surmise that this exon would probably tolerate the presence of a new intron within that exon. We don't have proof of this, but this is why we chose exon 3 essentially of that gene. So we chose that gene. Essentially, we chose SCYL1 also for a couple of reasons. The first one is that SCYL1 is a well-studied gene in our lab, and targeted disruption of SCYL1 causes phenotypic changes that are easily recognizable. These include growth retardation, as you can see here on the left image. It also causes an early onset motor neuron disorder, and that leads to the progressive loss of motor neurons and muscle weakness and muscle wasting due to muscle denervation, as shown here. This is the quadriceps femoris of a wild-type mouse, and this is the quadriceps femoris of a mutant mouse. So essentially, it's very, very easy to determine whether we have a loss of function phenotype. And this is going to become important, especially because we're trying to conditionally inactivate this gene using our new technology. The second reason is that we have all the reagents that are necessary to characterize these alleles. We have antibodies against SCYL1, and we have various mouse models, including SCYL1 knockout mice and mice with a conditional allele that has been engineered, oh my gosh, almost 12, 15 years ago by myself using conventional gene targeting in ES cells. And in this case, we flanked the entire gene with LOXP sites. So anyway, that was engineered a while back. So because we have these regions and because of the phenotype, that's why we decided to use SCYL1 as prototypic gene to test the applicability of this new technology for engineering mouse models with conditional alleles. So this is just the data. So from 2 rounds of microinjections, we obtained 12 mice with Cas9 activity. And by Cas9 activity, it means that we were able to see that in these mice, Cas9 had cleaved the target site. And 2 of these mice actually had the correct insertion, as shown here by PCR amplification and Sanger sequencing. This is the sequence that we obtained from the amplification of the locus. And as you can see, we can find the splice donor site, the LOXP site, the branch point right here in the middle, the polypyrimidine tract here in yellow, the second LOXP site, and the splice acceptor site. And this was fused. These little 2 junctions here indicate where recombination probably occur at the locus. So to test the functionality of the allele, we outbred mice to wild-type mice for a couple of generations, and we always do this, especially we always do this, and it's good practice to do this, to outbreed your new mouse models to wild type animals, to make sure that you get rid of additional mutation that you might have engineered, but also that you bring some fresh blood into your colony. So it's always good to maintain all your mouse models on the heterozygous background, cross onto wild type mice. All right. So we did this for a couple generations, and for each generation, these mice were characterized by PCR amplification, topo cloning, and Sanger sequencing. So when you engineer new mouse models, especially using CRISPR-Cas9 technology, you always end up with either chimeric animals or animals that have other mutations at the locus that you try to engineer. So you try to outbreed those mutations from your main colony. But you also want to make sure that the mutation that you have, or the newly engineered allele, is correct, so that you have to continue to characterize those alleles using not only just PCR amplification, PCR genotyping, but also Sanger sequencing and topo cloning. So from these crosses, we obtain F2 mice, and we these F2 mice together to produce homozygous mice, and this way we can test whether the allele or the conditional or the AIV4 cassette functions properly. These mice again were analyzed at the genomic level, the transcriptional, the transcript level, the protein level, as well as for phenotypic changes. And so what we found is that mice homozygous for the AIV4 allele, as shown here, expresses CYL1 to levels that are comparable to wild type animal on the left side here. So we have 3 homozygous samples from the AIV4 mice and 3 from the wild type allele on this Western blot here. So the expression was the same, not only at the protein level, but also at the RNA level, and consistent with the proper expression of a CYL1. Mice homozygous for the AIV4 allele actually showed no overt abnormalities, unlike the SCY1 deficient mice, which showed broad retardation, which we can measure by just weighing the mice. These SCY1 knockout mice also show muscle weakness, as you can see here, and then a motor dysfunction. And we do this by using a scoring system, but eventually they will become paralytic, like I showed you on the video. So together, these findings actually indicated that the AIV4 cassette is properly removed by the splicing machinery, leaving a functional transcript and normal expression of the gene. So this showed that actually we can knock in this small cassette within the exon 3 of SCY1, and the allele is still functional. So next, we wanted to test whether recombination between the 2 LOXP sites would occur despite their proximity. So these 2 LOXP sites within the cassettes are located 113 base pair from each other. So that's kind of close to allow the Cre-recombinase to actually recombine the 2 sites. So we wanted to know whether this was going to be efficient, and so we produce. What we did is that we crossed the CYL1 AIV4 mice, as shown here, with a Cre-deleter mouse, which we obtained from Jackson Lab. And again, these mice were characterized at the genomic level using PCR amplification, topo cloning, Sanger sequencing, so forth and so on. And what we found essentially is that that recombination frequency was near a little over 80% in the AIV4 mice, Cre-expressing AIV4 mice, as you can show here on the PCR genotyping stuff, but also the quantification here. And we wanted to compare this to a more conventional way where we have 2 LOXP sites flanking an entire gene. In this case, we also use the SCY1. Like I mentioned before, we have those conditional mice with this conditional allele. And as a comparison, what we found is that the recombination between these 2 LOXP sites are more distant than normal, but still, the recombination between these 2 LOXP sites occur in about the frequency of recombination is about 40% in these mice. So we were quite satisfied with this 80% recombination efficiency here, showing that even though the LOXP sites are very close to each other, it does not prevent the proper recombination of the allele. So finally, we wanted to test whether crippling the intron would result in the inactivation of the allele. And to do this, we crossed the CYL1 AIV4 mice that also express the Cre-regulated together with SCYL1 AIV4 homozygous mice. And from these crosses, we obtain Cre-positive SCYL1 AIV4 delta. We call the recombined allele the delta allele. And so again, these mice were characterized at the genomic level using the previous methodologies. And so from these crosses, we actually obtain mice homozygous for the recombined allele. As you can see here on that PCR genotyping, the recombined allele is the middle band here. Proper recombination of the cassette was actually confirmed by PCR amplification and Sanger sequencing, where you can see the sequences encoding the splice donor site here, followed by a LOXP site. And this was followed by the splice acceptor site. So we, as homozygous for the recombined allele, we were able to PCR amplify and sequence this, and we saw that the recombination occurred properly. And probably more importantly than any of this, we also found that that mice homozygous for the recombined allele, like SCYL1 deficient mice, which is the third line here, they failed to express the CYL1 as shown by this Western blot. We used two different antibodies, one that recognized a C-terminal segment of SCYL1, and the other one recognized the variant terminus of SCYL1. In both cases, we were able to find that the mice homozygous for the recombined allele showed no expression of SCYL1 and didn't show any truncated form of the protein. And that's going to become important in a minute when we explain a little bit how the allele works and how the expression of the gene actually is regulated. So not only did these mice fail to express a CYL1, but they also exhibited phenotypic changes consistent with the loss of SCYL1 function in vivo, and these include postnatal growth retardation. This is your homozygous mouse with the inactivated allele, and this is the control mouse obtained from the same litter. These mice also show that's growth retardation, and they also show muscle weakness, as did the SCYL1 knockout mice. And these mice also show motor dysfunction as assessed by our scoring system. And this is just to show you that these mice also exhibit massive muscle wasting. This is the leg of a mouse homozygous for the inactivated allele. And this is just a cross-section of the quadriceps femoris again, showing this massive atrophy. And what we also know from the SCYL1 deficient mice is that this massive muscle wasting is due to muscle denervation. So from these results, we actually concluded that the DECAI approach can be used to engineer conditional alleles in mice. The AIV4 cassette is recognized by the splicing machinery and removed to allow expression of SCYL1. Upon Cre-mediated recombination, the cassette is inactivated, preventing expression of SCYL1. The insertion of the intron, or the small cassette, is actually efficient and occurs in more than 10% of the mice that we engineer. When we did this first mouse model, we have been working on several other mouse models, and we typically get insertion rates of about 25%. So for this was based on 5 new mouse models that we are engineering as we speak. The characterization of the allele at the genomic level is kind of straightforward. It only requires one set of primers, PCR amplification of the locus, topocloning of those amplicons, and Sanger sequencing. Of course, you can also do direct Sanger sequencing, but it becomes a little bit more complicated to actually be able to read and assess whether the cassette was engineered properly. So that's why we use topocloning. Finally, the HDR template is easy and affordable to produce. So essentially what we use to engineer those alleles is a short 300-ish nucleotide long single-stranded DNA molecule, and that's all that's required. So it's cheap to produce, It's easy to manipulate, and the homology arms that we have used in this particular model were 16 nucleotide and 58 nucleotide on the other side. So fairly straightforward technology to engineer conditional alleles in mice. All right, now I'm going to get into the details a little bit more and provide some mechanistic insights into the way we believe the allele is being inactivated. So bear with me. This might be a little bit more less straightforward to understand, but I'll try to keep it simple for you. All right, so we believe that, at least in this case, the inactivation of the allele occurs not only at the transcriptional level, as it is, as it would be envisioned, if you will, or as we thought, at least it would occur, but it's also probably regulated at the translational level, but also as well as the post-translational level. And the reason for this and the explanation for this is actually based on the following observations. The first observation is that at the transcript level, as shown here in panel A, where we did RT-qPCR, what we found is the expression of SCYL1 seems to be the same in wild type and mutant mice. So the RNA transcript is not really sent out for degradation via the nonsense-mediated mRNA DECAI pathway. But what we found actually is that it's a little bit more interesting than that, is that what we found is that it is not necessarily the abundance of the transcript that's changed, but it's actually the nature of that transcript that's different. So using RT-PCR, topo cloning, and Sanger sequencing, what we found is that we observed 5 different transcripts that were produced from the recombined. So these shorter transcripts here were not expected at all. And these shorter transcripts, including the longer ones, essentially they represent, they're made of 5 different transcript variants that are all produced from the recombined allele. 3 of these transcripts, variant 1, 2, and 5 here shown in the upper right panel, were actually in-frame variants and resulted from the splicing of the AIV4 splice donor into exon 4 of the gene. The second resulted from the splicing of the AIV4 cassette within a cryptic splice acceptor site within exon 3. And transcript variant 5 was a result of a splicing from the cryptic splice donor site in exon 3 into the splice acceptor site of the artificial intron cassette that's recombined. So luckily for us, these variants actually all 3 of them produce very unstable proteins, as you can see here by the ectopic expression of those variants in HEC293 T cells, where you have very nice expression of the wild type form, but these 3 other in-frame variants actually were not stable, or actually expressed much less than the wild type counterpart. This is the quantification of this protein expression based on relative to the RNA transcript that were present in those cells. So the idea is that it looks like there are in-frame transcript that would express proteins, but these proteins are a lot less, either not as well expressed or folded, or actually maybe less stable. And we also did a kind of a half-life experiment using a cyclohexamide chase, and what we found also is that these mutant forms of SCYL1 were actually less stable than the wild type full-length SCYL1. So from these results, we concluded that the regulation of the expression of the protein not only occurs at the transcriptional level, but also at the translational and post-translational level. So that was kind of a surprise to us. And based on this, we came up with the following recommendations for the design of conditional alleles using this strategy. The first one is that the cassette should be inserted within the 5' most exon of a gene and within the 5' half of an exon of that particular gene to promote mRNA degradation via the nonsense-mediated mRNA DECAI pathway, as we proposed at first. What we also suggest is that the artificial intron should be inserted such that potential splicing events driven by the AIV4 splice donor site with downstream exon results in an out of frame transcript. So essentially, you have to position this small intron such that to make sure that if there is any splicing event between that splice donor site from the short cassette into downstream exons, this will result in the inactivation of the gene as well. So the placement of the artificial intron should be should not be limited to actually the availability of optimal sequences upstream of the splice donor and downstream of the splice acceptor site. This is a little bit more details than you would like to know, but the idea is that there are sequences that seem to be favored surrounding splice acceptor sites and splice donor sites. So, but in the positioning of these cassettes, it looks like it's not very important, because when we engineer this particular cassette, at first I did not even look into this, and we did not put the cassette within the most optimal sequence environment, if you will. And lastly, and it's not on the slide here, but it came out to me, it came up to me last night when I was preparing those slides, is that if possible, the AIV4 cassette should be placed within a gene segment encoding an important structural feature of that gene product or that protein. So if you have a kinase domain, maybe you would like to put that AIV4 cassette in the kinase domain, so that if there is a little frame shift, there are some mutation, then the protein that's produced would be actually would not be stable and be degraded. All right, so this is what we came up with for the in terms of recommendation for the engineering of these alleles, and now we are trying to put these recommendations into application. So so following our success in engineering conditional allele using this short artificial intron, we decided to see whether we could expand the applicability of this technology and make it like more universal, or we could actually establish this procedure as a universal approach to engineering conditional alleles. And so what we have done is that we have been targeting small genes with overlapping coding regions. For example, there's a gene running in the opposite direction here. So we think that it will allow us to be able to conditionally inactivate this gene without influencing expression of that other gene here. We also are working on engineering a mouse model actually with large in which that would target genes that actually have embedded other sequences, like a long non-coding RNA transcript, for example. We also believe that this strategy can be used to target single exon genes. So we have a project right now where we are targeting a gene that has only one exon. And we're also interested to see if we can apply this technology to genes that have multiple exons, and one of them has nearly 100 exons. So we're kind of curious to see whether engineered conditional alleles using this new short cassette strategy will actually work for those massive genes. We'll see what we get. We don't have data yet for me to show you. We're also working on developing artificial introns with site-specific recombinase systems like the FLIP-FRITZ system, for example, but we could also develop other systems like VicaVox and DREROX and so forth and so on, the ones that I mentioned at the very beginning. We believe that this could be used to engineer conditional alleles that could be crossed with mice also that have a conditional allele using the Cre-regulated system. So that could be very useful, but we have to show that actually this is working. We actually just generated the mouse model, I think yesterday. We have some data that suggests we might have it. So this is kind of exciting, and we're looking forward to characterizing this mouse. We're also developing strategies for the conditional activation of genes. And this is actually using what we call an inverted artificial intron. The cassette that we're using is called AIV6. And we think that we can use this to not only express conditionally genes, but also we can engineer conditional point mutations. So this could be very useful for cancer biology people that are studying point mutations that you can express, for example, a mutant form of a protein within a particular tissue and see the impact on I don't know, tumor formation, tumor development, and progression, and so forth and so on. So in this system, essentially, there's a small cassette that contains sequences encoding a LOX71 and a LOX66 site. These are oriented in the opposite direction, so head-to-head, if you will. And these are flanking an inverted intronic sequences, so such that the cassette cannot be recognized essentially as an intron and is not removed. So it inactivates the allele. And upon Cre-mediated recombination, this cassette is flipped, now allowing expression of the gene. And if you wanted to, you could introduce a small missense mutation here, and then you would have expression of a mutant form of your protein. So for those who are interested in this technology, you can go check out the BioRx or the Sneak Peek website. We published our paper on there. The paper is also been accepted by Helion, which is a cell press journal that publishes proof of concept experiments. So paper should come out pretty soon. We're actually just revising it right now, and there's just a few typos we need to correct. The final what else do I want to say here? No, I think that's pretty much it. So please just go check out the paper. It's fairly interesting, and it has a little bit more detail than I just give you. All right. I just want to take a few minutes to thank the individuals who participated in this study. This was literally a team effort. So I'd like to thank members of the IUGEC, which all of them took part or had a little part to play in the characterization and the generation of this model. So shout out to Annelise Cassidy, Destiny Thomas, Samantha Baer, Max Tubin, and Hanyan Chen. I would also like to thank former lab member Emin Kuliev, who actually started this thing with me in 2020, right before I joined IU. I would like to thank Purdue, Judy Hallett from the Purdue University Transgenic Core Facility, who has been working with us and help us engineering these new animal models with these conditional alleles. I want to thank people from the St. Jude Children's Research Hospital Transgenic and Knockout Core Facility, Hartmut Burns, Jack Subbett, and Xiuling Li, who actually generated the original SCY1AIV4 allele or knock-in. For those interested in our services, please visit our website or just Google IUGEC School of Medicine and you'll find us. You can also contact us. You can send me an email, or you can also get in touch with Annelise Cassidy or Destiny Thomas. I just want to say that we are currently recruiting. We are looking for a lab research assistant to help us engineer cell line models, particularly stem cell models. And yeah, so if you are interested or know anyone, the job posting is 304-114. So just tell them to go check it out. We are recruiting for this. We are also recruiting. We're also going to bring a new microinjectionist in to help us with the large knock-ins and things like that. So things are moving forward. All right, and with this, I'm happy to take any questions related to our core services, technology development, any questions. But I'll try to answer that. Thank you.
All right, thank you, Stefan. I can ask, I guess, a first one while people are getting their courage up. So is it available now? Are you taking projects from other investigators, or are you still kind of working out the bugs?
We have some pilot projects with some investigators, and we've been very clear that this is a new technology that we are developing. I think, we have 5, 6, 7 new mouse models with these conditional alleles. I wish I could tell you, yes, go ahead, call us, we'll make those alleles, but we cannot at the moment, just because we need to make sure that it's going to, we can apply this technology to many different genes. we're trying to speed this up a little bit, so we're cheating a little bit on in terms of crossing mice and everything, so just to speed it up. But at the end of the day, we hope that within 6 months we're going to be able officially to say, yes, this is a technology that can be used to conditionally inactivate any gene, and we know the secret of the recipe and everything. Yeah, but it's, again, it is working. It's worked really well for one of our genes. Now we are developing these other 8, 9 models, and hopefully soon we'll have great data to present as well with these models. Yeah.
So I was curious if people wanted to look at, let's say, a specific genetic variant or a mutation, you would have to engineer that separately from the intron. Is that what? Did I understand that correctly?
Right. So if you are interested in making like a conditional missense mutation, for example, then what we would do is that we would target that particular exon, introduce this artificial intron, and next to it we would introduce that missense mutation, so that normally the allele would not be expressed, and upon flipping of that little cassette, now the gene would be the cassette would be spliced out, allowing expression of the protein with the particular point mutation. And I think this is particularly interesting for cancer biology, where you don't want to have germline missense mutation that are driving tumorigenesis. You really want to introduce this at a later stage during development. And this goes for it. I'm just saying cancer biology, but it goes for any kind of protein or mutation that we're studying. If the mutation is not germline and it occurs later on during development, then that's kind of interesting strategy. So we've been working on this. It's like, again, this we have many projects. We have to deal with many different things. So these development, the technological advances are a little bit slower than I would like to, but we're getting there. So yeah. So it takes a village to actually get these things done. It's fascinating. All right, thank you all.